Hello students, welcome to EPG Pathshala. I am Dr. Sudhita Gautam from Hansraj College, University of Delhi. Today we are going to discuss about a module, Principles of Gene Expression, that is transcription, under paper Molecular Genetics. After studying this molecule, you will come to learn about the second part of the central dogma of molecular biology, the concepts of genetic codon, that is, a triplet codon, Wobble hypothesis, conversion of genetic code to polypeptide, RNA-directed protein synthesis, and the mechanism involved in protein synthesis. Translation, the process of protein synthesis. The central dogma of molecular biology explains that DNA codes for RNA, which codes for proteins. DNA is the molecule of heredity which passes from parents to the offsprings. It contains the instructions for building RNA and proteins, which makes up the structure of the body and carries out most of its functions. Translation is the second step of central dogma, which describes how amino acids are produced from the genetic code, a process where ribosomes synthesize proteins using the mature mRNA transcript produced during transcription. Proteins are the building blocks of body composed of amino acids linked together through peptide bonds. It involves binding of ribosome, thought of as a moving protein synthesizing machine near the 5' end of mRNA and moving towards the 3' end. Protein synthesis takes place through a series of sequential interaction of RNA molecules, mRNA and the rRNA that holds the mRNA in the ribosome. The way in which a sequence of DNA bases is transcribed into complementary RNA bases is then translated into corresponding amino acids takes place in the cytoplasm, and the nucleotide sequence of the mRNA is converted into the amino acid sequence of a polypeptide. Expression of genes is highly important for studying the flow of genetic information from DNA to proteins to fully understand the music of life. Need to explore the central dogma further. Translation. I'll climb up this ladder of DNA to see where life takes me. Translation is the second step of central dogma, which describes how amino acids are produced from the genetic code. Gene to protein. The sequence of nucleotides, namely adenine, guanine, cytosine and thymine, joined together in sets of 3 to code for 20 known amino acids. George Gamow proposed that each amino acid has 3 sets of nucleotide which are known as the codon. He concluded it by a bit of armchair logic, stating that number of words possible with 4 letters are 1, using all 4 letters, 16, 4 square, using 2 letters, and 64, 4 cube, using 3 letters. As there were 20 different amino acids, he concluded that each codon must be containing 3 nucleotides. A single base cannot code for one amino acid because this would give rise to only 4 amino acids. If we consider a 2-base code, it provides only 16 possibilities. Thus, a minimum of 3 bases is needed to specify a single amino acid. Francis Crick, Sidney Brenner, and colleagues at Cambridge University soon confirmed the findings of Gemau, that is, the triplet nature of codon. Because there are 61 sense codon, it was soon revealed that a single amino acid is coded by more than one codon. Two adjectives that are often used to describe the genetic code are unambiguous and redundant. Unambiguous means that the codons are fixed and that each codon specifies one amino acid. For example, ACC codon codes for tryptophan and nothing else. Several codons may code for a single amino acid that is redundant. For example, CAA, CAC, CAG, and CAT all code for a single amino acid valine. Characteristic features of genetic code. The genetic code is triplet in nature. 3 nucleotides code for a single amino acid. The genetic code is continuous. There is no break while reading the mRNA. mRNA is read as a comma-free stretch of 3 nucleotides without skipping any nucleotide. The genetic code does not overlap. The genetic code is degenerate. That is, each amino acid has more than one codon with an exception of AUG for methionine and UGG for tryptophan. The start codon or AUG and stop codons are UAA, UAG and UGA are same for both prokaryotes as well as eukaryotes. Sir Archibald Garrod, 1909, a British physician, first proposed that different genes dictate different phenotypes through enzymes. He proposed the idea that diseases came about through a metabolic route leading to a molecular basis of inheritance. George Beadle and Edward Topham together gave the one gene, one enzyme concept, for which they were awarded the Nobel Prize in Medicine or Physiology in 1958. According to one gene, one enzyme concept, a single gene is responsible for role of a particular enzyme and thus established genes as chemical molecules. Experimental evidence for triplet genetic code. The genetic code was cracked by Marshall Nirenberg and Johann Heinrich Mattei, 1961. They determined the codon sequence of bases for phenylalanine. Enzyme polynucleotide phosphorylase was used to create synthetic RNAs. Enzyme was able to link any RNA nucleotide without the need of a template. The first synthetic RNAs used by them were homopolymers, which contained only a single type of nucleotide. They tested a polynucleotide phosphorylase consisting exclusively of poly-U. Thus, they obtained a tRNA molecule consisting of entirely uracil nucleotides which had only U codons. These poly-U RNAs were added to 20 experimental sets containing all 20 amino acids and components necessary for translation. In each test tube, one individual amino acid was radio-labeled, which varied in each test tube. On observing the test tubes, they found that radioactive protein appeared only in one test tube, which contained labeled phenylalanine. Thus, it was proved that UUU codes for phenylalanine. Later on, codons were followed for lysine, glycine, and proline. Today, we know the sequence of bases which code for each amino acid found in proteins. Normal and Wobble pairing. Wobble hypothesis. Francis Crick in 1966 gave the Wobble hypothesis, according to which there is a flexibility of base at the third position of the codon. For example, alanine is coded by GCU, GCA, GCC, and GCG. One should note that all codon begins with GC, but the bonding at third position varies. This non-standard pairing is referred to as the Wobble base. Let us begin to answer how translation is carried inside the cell. To explore translation further, I need to look in for its ingredients and machinery within the cell. Translation ingredients. Three essential components of translation include: first, mRNA as template, tRNA as transporter of amino acid, and ribosome subunits as machinery to assemble the proteins. mRNA, it is the RNA molecule which acts as a messenger to convey the genetic information from RNA to proteins. Transfer RNA, RNA molecules having a clover leaf-like structure and transfer amino acids to the growing polypeptide chain. Ribosome, a site of protein synthesis and consists of two subunits, a larger subunit and a smaller subunit. Structure of ribosome. Ribosomes, machinery to assemble proteins, consists of two parts, a large subunit and a small subunit made up of rRNA and proteins. Functional ribosome is formed when the two units attach to each other. Each ribosome has three binding sites for mRNA. First, the P site, that is peptidyl site. 2, the A site, the amino acyl site, and 3, the E site or the exit site. Structure of tRNA. tRNA or transporter of amino acid. tRNA has a L-shaped tertiary structure and clover leaf-like secondary structure bearing 4 arms formed by the coiling of a single strand of 80 ribonucleotides. It plays the role of an interpreter between nucleic acid and peptide sequences by picking up amino acids which correspond to the proper codons in mRNA. Based on base-base complementarity, the second structure of transfer RNA can be drawn as the familiar clover leaf structure of four loops, each consisting of 4 to 7 Watson-Crick type base pairs. 5 regions of the tRNA are not base paired: the acceptor loop or the 3' end, the DHU loop named for the presence of dihydrouridine, the anticodon loop which interacts with the mRNA or the extra arm, and the TC loop named for the presence of pseudouridine base. Acceptor arm or 3' end. The amino acid which corresponds to the mRNA codon is attached at the 3' end of transfer RNA, which has the sequence CCA. The proper amino acid is joined to the tRNA by the enzyme amino-acyl tRNA synthetase. There is one type of amino-acyl tRNA synthetase for each amino acid, and the active site of each fits only the specific combination of the proper amino acid and tRNA. This binding of the tRNA and amino acid is highly specific, and the amino acid binds to the adenine nucleotide at the end of the tRNA. Anticodon arm present at the bottom of the loop, having 3 nucleotides or anticodon which are complementary to the mRNA codon. A single tRNA can recognize different codons for an amino acid. TC loop presence of pseudouridine base, and DHU loop presence of dihydrouridine base pair. Translational ingredients. 3 essential components of translation include messenger RNA as template, transfer RNA as transporter of amino acid, and ribosome subunits as machinery to assemble the proteins. mRNA as template. The mRNA codon recognizes the transfer RNA anticodon. Messenger RNA is complementary to the gene template and carries the genetic information from the DNA to the ribosome. The nucleotides of messenger RNA are read in a series of triplets which code for a corresponding amino acids and are referred to as a codon. Each amino acid has a specific codon. The genetic code consists of triplet continuous, non-overlapping and universal codons. This code is redundant or degenerate because a single amino acid can have more than one codon. The start and stop codon are same for all the amino acids. AUG is the initiation or start codon. 3 stop codons are UGA, UAG and UAA. How does these ingredients carry on with the entire process of translation? A bigger question, mechanism of translation involves 3-step processes. First, initiation. Second, elongation. Third, termination. Steps of translation. First, initiation, binding of ribosome, transfer RNA and messenger RNA, elongation, peptide bond formation between two amino acids and to repeat between two amino acids to the growing polypeptide, termination, release of the polypeptide and breakdown of the entire machinery. Charging of transfer RNA. The amino acid and ATP bind to the specific amino acyl transfer RNA synthetase enzyme to give amino acyl transfer RNA. Amino acyl transfer RNA synthetase binds amino acid to transfer RNA molecule. There are 20 different amino acyl transfer RNA synthetases which are unique for each amino acid. Each synthetase is able to recognize its own unique amino acid based on its different shape, size, charges, and R group of the amino acid. This reaction takes place in two steps, utilizing the energy from an ATP molecule. First, the amino acyl AMP enzyme binds to the uncharged tRNA. The amino acid is transferred to the transfer RNA accompanied with the release of AMP molecule. The enzyme returns to its original configuration and is ready to bind with next upcoming amino acid and ATP molecule to start the process all over again. First step, amino acid and binds to the enzyme. Enzyme catalyzes the coupling of amino acid to AMP to form amino acyl AMP. Two phosphates are lost in the reaction. Amino acyl AMP enzyme and uncharged tRNA bind to each other. Enzyme transfers amino acid from amino acyl AMP to tRNA to form amino acyl tRNA or AAtRNA. The AAtRNA and AMP are released from the enzyme. AAtRNA and AMP are released. Enzyme returns to the original state and is ready to bind to the new amino acid. Why is charging of transfer RNA required? How does first initiator transfer RNA differ from other tRNAs? Is there any difference between the initiator transfer RNA of prokaryotes and eukaryotes? Let us find the answers. Initiator T transfer RNA uniqueness. The first amino acid, methionine, varies with the subsequent methionine which are added in between the growing polypeptide chain. Starting amino acid of prokaryotes is N-formyl methionine transfer RNA and of eukaryotes is methionine transfer RNA. The first codon is AUG, which codes for methionine residue and is referred to as the transfer RNA F methionine. Deformalization blocks the free amino acid and prevents the methionine to participate in the elongation reaction. It can recognize the reading frame for translation to begin. A series of 3 GC pairs in the stem that precedes the loop containing the anticodon is unique to tRNA F-methionine. These base pairs are required to allow the F-methionine tRNA to be inserted directly into the P site. Thus, it binds directly to P site of small ribosomal subunit. Initiation involves an mRNA molecule, a ribosome unit, initiation factor, initiated tRNA charged with N-formylmethionine and GTP. The small subunit 30S of the ribosome binds to the upstream of the 5' site of the AUG called the ribosome binding site or RBS. This messenger RNA RBS in prokaryotes is commonly known as the Shine-Dalgaro sequence. This pyrimidine-rich region pairs with the 3' end of the 16S RNA, part of the small subunit of ribosome. If a mutation occurs in Shine-Dalgaro sequence or the corresponding 16S rRNA, it results in failure of pairing for a particular mRNA. As a result, the process of translation does not take place for that particular mRNA. No corresponding sequence occurs in the eukaryotic mRNA. Initiation of translation. The small subunit of eukaryotic ribosome, along with the initiation factors, recognizes the gap at the 5' end of messenger RNA, which plays a crucial role in the initiation of translation. Initiating factor 3 or IF3 prevents the binding of the larger subunit of ribosome prior to mRNA binding. The complex of initiating factor 2 or IF2, GTP and transfer RNA charged with N-formylmethionine binds to small ribosome subunit along with initiating factor 1 or IF1 to form the 30S initiation complex. In the final step of initiation, IF3 is released, which facilitates the binding of large subunit of ribosome. The charged transfer RNA on the P site is still attached and the larger ribosomal subunit joins with the smaller unit, giving rise to the 70S initiation complex. Hydrolysis of GTP gives GDP and PI and releases initiating factor 1 and initiating factor 2. This initiation complex proceeds downstream in 5' to 3' direction until it encounters the start codon AUG. The region between the messenger RNA cap and the AUG is known as the 5' untranslated region or 5' UTR. The recognition of the start codon in eukaryotes is facilitated by presence of a consensus sequence known as the Kozak sequence, sequence 5' ACCAUGC 3'. Initiator tRNA is the only tRNA which can directly bind at the P site. The P site is so named because, with the exception of initiator transfer RNA, it binds only to a peptidyl transfer RNA molecule, that is, a transfer RNA with the growing peptide attached to it. Amino acyl tRNA binds to the ribosome and forms a peptide bond between the two amino acids. The translocation of tRNA from P site to E site is followed by the release from E site and binding of a new amino acyl tRNA at the A site. It helps in binding the polypeptide chain one by one with the corresponding amino acid as deciphered from the template mRNA. Elongation. It consists of three prominent steps. First, binding of ribosome unit with charged transfer RNA, an amino acyl transfer RNA, a tRNA covalently bound to its amino acid, is able to base pair with the next codon on the mRNA arrives at the A site. This charged tRNA is associated with an elongation factor called elongation factor Tu in bacteria and elongation factor 1 in eukaryotes, and GTP, the source of the needed energy. Binding of the tRNA at a site results in GTP hydrolysis and release of elongation factor Tu. Elongation factor Ts helps to regenerate elongation factor Tu GDP to elongation factor Tu GTP. Second step, formation of peptide bond between the amino acid. The ribosome maintains the correct position of the amino acids present in transfer RNAs so that a peptide is formed between them. The bond between the amino acid and the transfer RNA in the peptidyl site or P site is cleaved first. Secondly, a peptide bond is formed between the released F-methionine and amino acid attached to transfer RNA at the A site. Once the bond is formed, transfer RNA at P site becomes uncharged and the transfer RNA at A site has the first two amino acids. Formation of the peptide bond is a result of the catalytic activity of the ribosomal RNA in the larger subunit of ribosome. Thus, this ribosomal RNA acts as a ribosome. Translocation of ribosome along the messenger RNA, one codon at a time. Movement of the ribosome along 5' to 3' direction of the messenger RNA positions it over the next codon, where a new charged transfer RNA can come and bind. As the ribosome moves downstream of messenger RNA, the transfer RNAs at peptidyl site and A site are still attached to the messenger RNA through the codon-anticodon pairing. The shifting of ribosome moves the transfer RNA from peptidyl site to E exit site and from amino acid site to peptidyl site respectively. This shift also vacates the A site, which is now ready to receive a new charged transfer RNA as specified by the codon. Termination of translation is signaled by the stop codons, which are UGA, UAG and UAA. Once the ribosome recognizes the stop codon with the help of release factors, it can trigger the release of polypeptide from the tRNA. Release of tRNA from the ribosome and dissociation of the ribosome complex. Translation stops when the ribosome encounters a stop codon. Translation termination. There are no transfer RNA molecules with anticodons for a stop codon. No transfer RNA enters the A site. Releasing factor or RF1 binds at the A site, whereas releasing factor 3 coupled with GTP attaches to the larger subunit of ribosome. The release factor RF1 recognizes the stop codons UAA and UAG, whereas the release factor RF2 recognizes UAA and UGA. A third release factor RF3 is also required for translational termination. The polypeptide is cleaved from the transfer RNA in the P site along with the hydrolysis of the GTP. The ribosome splits into its subunits, which can later be reassembled for another round of protein synthesis. So students, let us summarize what we have learned in this module. We have learned that translation is a process of protein synthesis which makes use of messenger RNA produced by transcription. It takes place in the cytoplasm. The genetic code is a triplet codon which guides the binding of individual amino acids. Genetic code is degenerate, non-overlapping, universal and redundant. Translation takes place in 4 phases: charging of tRNA, initiation, elongation and termination. AUG is the first amino acid. It binds to the tRNA with the help of enzyme amino acyl synthetases, making use of GTP as the energy source. This charged tRNA is referred to as the initiator tRNA, has unique property to bind to the P site. Prokaryotes have N-formylmethionine or F-methionine as the first amino acid, whereas in eukaryotes it is methionine. First, 30S initiation complex is formed, from which release of initiating factor 3 vacates the site for 50S or large ribosomal subunit to form the 70S initiation complex. Elongation proceeds in the 3' direction. An incoming charged tRNA attaches to the vacant A site. A peptide bond is formed between the first and second amino acid, connecting the amino acid of the tRNA, and the tRNA shifts from the P site to the E site and A site to P site respectively. Incoming tRNA binds at A site. This pattern continues and the ribosome translates the mRNA molecule until it encounters a termination codon on the mRNA. Termination takes place on encountering the stop codon, which are UAA, UAG and UGA. When a ribosome reaches a stop codon, the A site accepts a protein called a release factor instead of tRNA. The release factor breaks the bond between the tRNA and the polypeptide. As a result, the growing polypeptide chain is released from the tRNA molecule and the entire machinery dissociates. Thank you.